In this video, we will cover the CDG or congenital disorder of glycosylation that are related to Mannose or that will cause problems in the Mannose related glycosylation. So, we will cover three of those CDGs. First one is the Mannose phosphate isomerase. So, in this kind of congenital disorder of glycosylation, what happens is that this ability of the cells to convert fructose 6 phosphate to mannose 6 phosphate is weakened or is impaired. So, because of this, because of this, what happens is that there is a lack of this mannose 6 phosphate to the cells that will leads to abnormal glycosylation within the cells. So one of the therapy approaches that is suggested, or therapy ideas that is suggested, is to inject this mannose is to directly inject this mannose 6 phosphate to the patients so that they can overcome this barrier, that is mannose 6 weakened mannose 6 phosphate isomerase or impaired conversion of fructose 6 phosphate to mannose 6 phosphate. This thing can be overcome. This barrier can be overcome by injecting, by directly supplementing, providing this mannose 6 phosphate to the patients. So this idea is similar to the ideas that we have discussed in the previous video. Next one is this abnormal functioning of phosphoglucomutase 1. So what happens because of this is that this glucose 1 phosphate to glucose 6 phosphate conversions get weakened, and because of that the further processing will get affected, and the cells will observe a weaken this GDP1 phosphate. So in order to overcome this, one of the approach is to provide this glucose 6 phosphate directly via oral injection. Or other approach is that as it is this it functions in both the sides. Another approach is that the patients that are in which this phosphoglucomutase 1 has been observed have found that there is a lack of UDP galactose. There is a lack of UDP galactose. So in this case, what can be done is that the direct oral injection of D-galactose could overcome this barrier. Thereby, those patients will be able to get normal post-translation modification as that of a healthy human beings. So this direct injection of D-galactose or oral injection of this D-galactose can be one of the therapeutic ideas, one of the approach to overcome the problems because of the weakened phosphoglucomutase 1. The last one is that this phosphomannose 2 or mutation or abnormal functioning of phosphomannose 2. This we have covered in our in some of our videos also. So what happens in this case is that the ability of the cells to convert this mannose 6 phosphate to mannose 1 phosphate gets weakened because of the mutation in PMM2 that leads to abnormal structure of this PMM2, which is not able to perform its function properly or efficiently. So in this case, there are some therapies that are already under therapy ideas that are already under investigation. So one of those therapeutic idea is to intravenously inject this mannose 1 phosphate through this lipidylic layer or dissolvable layer, in this dissolvable layer, so that cells will not face the scarcity of this GDP Mannose that finally goes into the endoplasmic reticulum for the glycosylation, or we can say for the initiation of asparagine linked glycosylation. That most of the proteins, or around more than 50% of the proteins, have this this kind of glycosylation. So abnormality of this PM2 will affect most of the proteins. So this strategy can be used, that is, intravenously inject mannose 1 phosphate in a lipidic layer or dissolvable layer, so that cell will not face the deficiency or scarcity of this mannose 1 phosphate, which can later be converted into the mannose GDP, and then be used for glycosylation in the endoplasmic reticulum. So these are three CDGs that are covered in this video that are related to Mannose. And if you need to understand this thing in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.